Okay, so since I have the microphone, so I get the first opportunity to speak, but I think we had a very beautiful diversification in the talks and usage of many different types of approaches to see really what makes us human and how we're diversifying from our ancestors and what are the using many different techniques and different approaches. So I think what I wanted to ask first is maybe what do you see in the future? Like, is it going to be more of, you're going to look more cells, more different animals? What do you think is the future in these questions?
Anyone want to volunteer to talk about?
At least one aspect of the future, I think, would be more combining. So, I mean, we're already going towards that. So combining DNA and RNA and lipids, metabolites together, and also more spatial analysis and everything to really get a better view of the phenomena with measuring.
So I can comment about the future from my perspective, which is that of ancient DNA and maybe, you know, Neanderthal cognitive skills. So until the day that we find a frozen Neanderthal with all the tissues intact, we will have to work hard in order to infer, to extrapolate DNA methylation from bones to brain and other tissues, and with relate to what Naomi said regarding combining. So this also goes for the field of ancient DNA. Work on ancient proteomics is already very much progressing, and there is a lot of new findings in this. And if DNA survives up to 1 million years, then some kind of proteins may survive in ancient tissue for much more. I don't know about lipids, if you can see anything in ancient DNA, but maybe you know. But indeed, combining different kinds of evidence would definitely be the solution.
Well, I have to say, if we want to see development of what makes us human, we need to a little bit change attitude towards it, because, for instance, research done using monkeys, it's something very, very disputed in many places. But if we want to understand what makes human brain different from brains of other primates, you have to look at other animal systems, because of course you can create, and people do create, wonderful model systems. But for instance, one thing what we need to integrate also is brain is a very complex organ. We cannot look at just how we do it, some specific parts to understand how it works. And I think tomorrow and the day after tomorrow there will be talks about how people try to integrate activity of the brain on from different regions, but we also need to do it on the molecular level. But for this, we need the brains. And if we continue to, you know, work with just rodents or just with some model systems, it's really hard to understand how brain works. We have to make our hands dirty and do some things with a real human and monkey brain. That's what I think.
Well, I sort of was trying to stall to get more time to think about this, and still don't have, and I still don't think I have anything very interesting to add to what my colleagues have already proposed. In terms of the DNA sequencing and RNA sequencing, I agree with Naomi that I feel like we're still in a fairly rapid growth phase where the technology is still advancing in terms of single cell methods, both at the RNA level and also at the DNA level, even for whether it's ancient DNA or even the genome of a single cell, comparing it to the genome of another single cell. And a lot of this is limited by cost, and so it's always hard to predict the future of anything, but but I think a lot of how fast we can progress at advancing a lot of these things is going to depend on Illumina and how fast or some other competitor and how fast the cost of sequencing drops, because that really enables a tremendous amount of stuff. In principle, it's theoretically possible to sort of take the brain of any person or any animal and sequence all the genomes of all the single cells and to deduce the entire lineage map of the whole brain of an individual, except obviously the cost is beyond imagination. But if cost were not an object, it's amazing what the sorts of things that could be done both at the RNA and the DNA level.
Okay, so my turn. Well, actually, I wanted to raise like another idea because, well, in my view, we're in science, we're trying to catch up with the science fiction, and I think all the time when I'm reading science fiction stories that were written 10 years ago, I say, well, as a matter of fact, I can do it today. So what's the point? So the question is, how will it change what we're doing today, how we're taking into consideration the science fiction of today into our studies? Like, I think, I want to ask you if you think, for example, today we have a lot of incorporation of artificial intelligence or machines within people that are taking over, or like, today babies are born with already the ability to use a touch screen at birth almost. And well, I have a 6-month-old grandson that is already doing like this. And it's completely intuitive. So I wonder how will it really change, like how the people will look at it in a million years? Because apparently, like the selective disadvantages or selections that we had in the past are nothing that we consider of. Like usually people, well, at least in the Western society, don't die of hunger, don't die of many other problems that we had in the past. So do you think it will change how the human will look like in a million years? Or
So, right, so that's a fascinating thing. You know, so evolution basically, I guess, again, is in an oversimplified way, it depends upon variation and selection. And so I guess the question is, you know, I'm always reminded of that first episode of the original Star Trek where there are those beings with these humongous heads. And of course, you know, in the case of humans, it's a little hard to imagine how we could evolve such humongous heads for a couple of reasons. First of all, because that would require some form of natural selection, and also secondly, because those beings would have to make it out of the birth canal, right? And so the evolution of our brain, presumably is some sort of dynamic equilibrium between the fact that it's good for humans to have a big brain because it generally makes you smarter, but it's bad for humans to have a big brain because then you can't make it out of the birth canal. And so I personally wonder whether now that C-sections actually make up whatever 20–25% of all births, whether this is actually removing a form of negative selection on human brain size. And so that hasn't, and if that becomes increasingly common, perhaps that could lead to humans looking like those guys from Star Trek in a few years. I just don't know, but it would seem to be a form of natural selection which is actually being altered. And so I suppose there's other things we could think about, but that seems to be one that comes to mind.
Yeah, okay, so So the pace of evolution is very slow, and we are trying really hard to look at what happened in the really recent hundreds of thousands of years, and we see very slow processes like shrinking of the brain. So it's apparently smaller than what it used to be for early anatomically modern humans. So indeed, these are all complicated processes involving both variation and selection, and also, like I think is one of the conclusions from the talk that I gave is that some of them are pleiotropic effects of genes, meaning that genes do a lot of stuff. They are involved in many systems in the body, and sometimes selection pressure towards one direction affects other direction. For example, the fact that the face went backwards and the larynx went downwards, they seem to be affected by the same genes. We have no clue if at all it involves selection and on which of these processes the selection acted, if at all. So all these processes are very complicated and very difficult to assess. Regarding science fiction, the literature in the field of ancient DNA is full with such ideas like, well, half science fiction, like returning mammoth to the world, which of course awaits for the development of genome editing technology to be able to run in high throughput, but these are ideas that people may start to toy with.
So actually your question took me in quite a different direction, maybe less science fiction-y. But the first thing that came to mind was how it actually will affect our social brain, like the transition from a more real world to a virtual world from such a young age. So I think, we see this effect in our society, but wondering also how it will affect kind of long term on our brain, and I think it will definitely affect the structure. But also my second thought was kind of more focused on my current focus, I guess, was that I think we'll live longer, and I think that we will maintain our quality of life, though, for many more years, hopefully. So that is also something kind of to take into account, to rearrange our society in a way as well because of these issues. And the first thing will be to really tackle the aging like how it affects our brain, but also in our entire body as a whole. So that was one thing I thought will be changing.
Well, I wanted to say I'm not, actually, I personally not very optimistic about, because I want to explain, because, finally everything happens like age and diseases, diversity in the population. It's all artifact of what we are, product of evolution, and evolution is a process which is very crude, and cruel. And it's a chance process where most species and individuals don't survive. And you also have to be replaced by new ones. That's why we all have limited lifespan. So everything we do is, by definition, to exempt ourselves from this kind of natural process. But in order to exempt ourselves, we need to take full control of our biology. And this is not yet possible. And even if it would be possible, it's not clear to me this could be done with adult individuals. So basically, if there is such a future, it's a very different future from what it is now. It's not something like we are all happy society, just taking some medicine and living longer. This could happen, but to a very limited extent, I think. And I think basically if there is a future which humans started to control their own biology, it would be a very different human society as it is now.
So we'll be happy to receive some questions, comments from the audience.
I don't think I need a mic if you were.
Okay, I can go.
One of the worries that I have with this more and more data, both of you mentioned that we're going to get more data, and of course that's true, is this feeling in genetics and the neurosciences that we're actually kind of drowning in data. I mean, you look at the early GWAS studies where, you get these candidate genes and then it wouldn't hold up. And now we've got even more data. We've got more candidate genes. We've got more candidate lipids. Now we've just seen another giant haystack that we need to go through. And I guess our problem is when we ask about what makes us human, we're looking for these few needles in the haystack that are the differences that make a difference, right? And I don't feel like the strategy of getting more data is going to help us solve that central problem. How do we find the needles? And I'm wondering if you guys have any insights. I mean, obviously it's hard work, but I mean, is there a fast track to finding the needles in the haystack?
So, did you want to answer? No. I guess I'll take a shot at that. But I think I know or sympathize a little bit with where you're coming from. An example is genome-wide association studies of schizophrenia, which initially were unproductive, and then the sample size grew, and they were able to find a few loci in the genome, and that was tremendously revolutionary. And then they got larger sample sizes, and they got up to a few dozen loci, and now they got up to really large sample sizes of like hundreds of thousands, and they're up to hundreds, if not thousands, of loci. And so when we've transitioned from dozens to hundreds of loci, have we actually learned anything more about the pathophysiology of the disease? So I think that's something about which reasonable people might disagree. I think, though, one way I conceptualize it is that these various loci have different effect sizes. And I think it's reasonable to say that those mutations that have very large effect sizes are often the sort of low-hanging fruit to try to understand pathophysiology. They're often the ones that are better modeled in animal models because there might be coding region changes or something like that. So I do believe there's a way of sorting through some of that complexity by looking for fairly simple things. We don't necessarily need to know every single locus in the human genome that makes us human. We could probably learn a lot about it by studying those loci which are of the largest effect. But I do sometimes also have the nightmare that someday we'll understand exactly how the brain works, but we won't. A computer will, but we'll actually not have the intuition to be able to understand what the computer is talking about when they're trying to explain it.
Yeah, I mean, to continue what Chris said, when we see that in like the single cell transcriptomics data, that when we increased our sample size we started with like 1,000 and now we're at hundreds of thousands, and even there are data sets of millions out there, is we actually do improve our classification quite significantly. And this is before taking into account what I just showed at the end of my talk about the inter-individual diversity, which is also more related to the GWAS and schizophrenia example. So I do think that we didn't get to that place yet where we have too much data. It does feel like we're drowning, that's true, but so we need maybe more computational people to help analyze the data, but I think that the system is very complex. So we do need, but of course, if there's a good way if we knew where to look, we didn't need this huge amount of data, that's for sure. But I have less ideas about that.
So I'm computational biologist, so I do not deal with generating data, just with analyzing it. So basically data is favorable and it is true that we are drowning with data, and there are new data science programs in Israel in different universities, and I had a chance to talk, and I tell the students, okay, that's good, now there is work for you when you grow up. So there is much more data than we can analyze now, that's for sure, and but I think, as it has been said before, it just adds to your power to detect lower effects and combinatorial combinations of effects. So it just adds to your power. It's more difficult to do. With regard to ancient DNA, we still don't have too much data. So if you go to really archaic humans, we have 3 individuals published that are highly covered, which is not nearly enough. And the number of high coverage samples that you can use for the kind of reconstruction of epigenetic signals, like I've shown in my talk, is still low. but there are also lots of works on SNP arrays data. And here we are already crossed the 1,000 sample barrier of different humans from across the world. And what you see, so it's like really accelerating during the past, I would say, 3 to 4 years. And what we see that with every new work that is published, you rewrite the history that you know. So these kinds of works mainly focus on understanding the more recent history of humans, and you do answer lots of questions that were open until not long ago.
Okay, so I also want to say something about that. So for take an example of schizophrenia, I think there is two levels of data. One thing, there are non-biological data we should not forget about. So maybe in 1 or 2 years, people will be able to predict people with schizophrenia with 100% accuracy just based on the things they buy online. This is very likely. And then all these genome-wide association studies might simply become non-funded anymore because what's the point? But from the biological perspective, actually, maybe surprisingly, I don't think we have enough data simply because every time we look deeper, we discover new things. Like some years ago, we didn't know there were microRNAs or other regulatory RNAs. Now, with a single cell, we see cell types which we didn't know existed. The same with optical microscopy, with other things. So actually, to create explanatory models, we need to know about the components of the systems. But we don't know all the components at all, and we don't know anything about many of the interactions they have. And we don't know them because there are some things which not even covered by existing technologies. So that's partially why we started to look at lipids, creating with another haystack, because this is not covered. But lipids is something very simple, and on the surface there are many things we don't understand about such complicated system as brain. So I think we need more technologies which would allow us to see different things in the brain and other organs to understand how things work.
So maybe adding to this, I wanted to add another maybe aspect, but actually today we have integration of social media with genetics. So there was recently an article done by Yaniv Ehrlich that he made a pedigree of 3 million people just using available data. And today you have tens of thousands of people that are sequencing their own genome, and many of them are willing to contribute and share their data, share their phenotypes, share their activities, and I'm sure that we'll be able to derive many new aspects just from this huge sampling that actually as much money that we can get to cover our research, you cannot get yet enough money to sequence hundreds of thousands of human beings at this stage yet. So I guess the novel ways of integrating existing data and contributing this data, new ways to analyze it, in addition to new methodologies that we cannot think about these days, will probably, explode the landscape of what we can learn, but also our abilities to extract data from it. Any additional questions that we want to raise, discuss? Yes? Can you transfer?
Considering that mice and rodents in general are in a significant different branch in the evolution of the brain, would that have implications on the significance of research that's been done on rodents, in terms of, like, would it be relevant, to invalidate the chunks of research that have been based on rodent research?
There's lots of good science. There's lots of great science on rodents that's safe and isn't going to be affected by this. There's also lots of poor science on rodents that didn't need this to be invalidated. I think particularly for the issue of brain size. I think you have to, I think now that we have the ability to do genome engineering on essentially any species, we can think a lot more imaginatively than we've been able to in the past about which animal is appropriate for this particular question that you have. And so for many questions, in fact most, rodents I think will continue to be the animal of choice just because they offer so many experimental advantages in terms of ease of manipulation and management and all sorts of animal care concerns and so forth. However, the door has really been opened, I think, to all sorts of novel species that if we can find ethical ways to do that we can consider as models that we might not have thought of before.
Okay, so
Yeah, I've often wondered about that. I mean, first of all, consanguinity is not necessarily a serious problem for humans. It doubles the rate of birth defects, something like that, compared to non-consanguineous couples. But in fact, that's comparable to having children when you're 40 instead of having children when you're 25. And nobody tells people not to have kids when they're 40. And so, it's debatable whether it really should be discouraged or not, as long as it's done as long as. But anyway, to get back to, I sometimes do worry about that in terms of, using laboratory mice to study behavior is a little bit like taking two humans and putting them on a desert island and having brother-sister matings for 100 generations, and then studying just those people who are too stupid to escape from the island, and using them as a model of humans. Because the laboratory mice are also, again, intensely selected to be too dumb to run away from their cage. And if they run away from their cage, we just let them go and we use the ones. And so I often wonder what sorts of genetic specializations mice have developed in order to be so tame and to live so well, and how that might. But again, there are lots of thoughtful ways to use. Again, this by no means invalidates all mice research, but it's something that's maybe worth thinking about a bit more than we have.
So we can observe, and you said it also, that the dynamics and the development of genetic tools is really amazing, yeah? And we are getting more and more technologies which will of course help to provide better diagnostic tools, which help to identify mechanisms of brain diseases, which is of course important. But I mean, the other side is of course that it goes so fast, and we are elucidating some mechanisms which might be very distinct, but cannot understand, so to say, the whole framework of interplays that we have when a certain gene type is changing. And we are not necessarily in a situation that we can predict for a patient, for instance, what does it mean if there's a certain gene defect. Yeah? So we may have a knowledge, but not necessarily have the tools to use this knowledge. And the question is how to make sure that in an ethical and informed way, we on the one hand go forward and solve our questions with respect to the methods, tools, tells, and so on. But on the other hand, do not forget that we speak about human beings, about patients, about society. Many of the effects that you are studying come out only in the second or third or next generation, not in this one. So these are really far-going methods that we have in our hands, and how to make sure that we on one hand not slow down the process of research, but on the other hand have an ethical, responsible handling of the tools, the genetic tools.
I mean, you're bringing a very important point about the ethical aspects, especially when we come to, I mean, we see how much we can learn from just DNA, RNA, DNA methylation, sequencing. We can learn so much about the individuals. So in the way, once the information is out there, we're exposing some very private information about the donors. So of course that's important to put in place, and there's very different ways to deposit, human versus like mouse data because of that. I think there's still a lot to think about how to do it better, and but currently I just see that as a great novelty of the donors. Because without their donations, we wouldn't be able to make any progress. And it's been an incredible thing to do, especially the longitudinal studies where people sign up quite early in life. They've been monitored, and after their death, they donate their brain or maybe also other organs to science. And this really makes such a huge difference to our studies. So I always kind of take the time to thank them just because I know it's not trivial at all, and there is some risk kind of involved in, and we should take the time to make sure we're handling the gift we get with careful gloves, if that's a saying in English or not.
Well, I think, things are changing so rapidly. Now with everybody has cell phone, and it means then everybody who has access to your cell phone knows when you go to sleep, where you go to sleep, and with whom you go to sleep, given the other person also has a cell phone, which is in most cases true for most of places. So, I mean, things which seemed incredibly private just maybe a decade ago, now something what still private, but in principle not anymore. Not anymore, and things on genetic level are much less easy to decipher than our behavioral patterns, which we basically make public by using this kind of gadgets and applications and everything else. So I think these concerns, of course, they are valid, but given other data which is becoming available about every one of us, including all these cameras on the streets you have and face recognition programs, which basically, even if you don't have cell phones, anyway, everybody can know where you are and where you went and what you bought with your credit card in the absence of cash. So I personally think we have to realize the society and is changing so rapidly. So being concerned about somebody looks at our DNA, this is the least of concerns I have.
Okay, so I think I'd like to close this session, and we're all ready for lunch.
Yeah. So really, thank you very much for all the morning speakers.